Hello, my name is James Spudich. I'm from Stanford University, and I'm very excited to tell you part 2 of a brief history of muscle biology. So in this part, we're going to be dealing with the period 1969 to present day, and the focus is going to be on the swinging Crossbridge hypothesis, which is what we ended with in part 1 of this series. So this is where we stopped from part 1, with Hugh Huxley proposing in 1969 the idea of a swinging crossbridge that went from one configuration, the crossbridge being about 15 nanometers long. By the way, that myosin head is called subfragment 1, or abbreviated S1. Goes from that configuration to a rotated configuration that would move the actin filament about 10 nanometers. So myosin can be divided into 2 parts. You can do this proteolytically in the laboratory by taking myosin and cleaving the coiled long tail in a position about here to give you a head domain which is soluble and has ATPase activity, and a tail domain called light meromyosin, which is insoluble and assembles into things that are somewhat resemble a thick filament. So the LMM is what's responsible for making the thick filament, and the HMM is really the part of the molecule that acts as a motor and has the ATPase activity. Now, a very famous and critically important experiment, which was done in 1971 by Lim and Taylor, who were consummate biochemists, and were using the new technology, relatively new technology, of being able to look at millisecond time resolution at events in biochemical reactions. And they used this to look at how fast myosin hydrolyzes its bound ATP to ADP and phosphate. And they got a very striking result. The striking result that you can see here is that the myosin alone, which is the dark circles, hydrolyzes within just a few milliseconds the ATP, even in the absence of any actin being around. And since HMM has 2 heads, what they found was they got about 2 moles of ATP, 1 per head, that was hydrolyzed, and then nothing much more happened. It was flat. So it was clear that the myosin alone had the capability of hydrolyzing the ATP, but it could not let go of the ADP and phosphate, which remained in the active site. And that when you added actin, that didn't really change the rate of the hydrolysis of the ATP, but what it did was gave you a steady state increase that kept going up the more actin you added, showing that what actin was doing almost certainly was activating the release of the products, so another ATP could bind. So this was really an incredibly important experiment, and led to kind of a combination of the chemical and mechanical ATPase cycles, which is shown here. So here's the chemo-mechanical ATPase cycle, where the myosin alone can bind ATP and hydrolyze it very quickly to ADP and phosphate. We now know that that hydrolysis causes the head to change configuration from a post-stroke state to a pre-stroke state, getting it ready to go on and do its cross-bridge motion. On the other hand, I haven't told you how we know that that works yet. That's going to come as we proceed. But when this head is cocked in the prestroke state, the model says now there's a slow step where it has to bind to the actin. And once it binds to the actin, that triggers the release of phosphate, where you have a concomitant power stroke giving you this putative 10 nanometers or so of movement. And phosphate release followed by ADP release. And once the ADP comes out of the myosin pocket, now ATP can go in, which changes the conformation of the head in some way that allows it to release from the actin. And now you're back to where you started. So that was the chemo-mechanical model that people had in mind in the early part of the 1970s. Okay? But what you may not know is that between 1969 and about 1980, the mid-1980s, this model was actually falling out of favor. And it was falling out of favor because really terrific biophysicists like Roger Cook and David Thomas in particular, who were independently and sometimes together putting biophysical probes onto the myosin head on a particular active, reactive sulfhydryl group, and then looking at these probes which were designed to be able to sense the orientation of the head. Okay? And they failed to see the expected pre- and post-stroke states. Many different kinds of experiments. And maybe even more troubling were really very challenging experiments taken on by Toshio Yanagita in Japan, a very good friend of mine, although we have been competitors scientifically for many years. In Toshio's really elegant studies, but complicated studies, he found that it looked like you had much more movement per ATP hydrolyzed than the 10 nanometers that you would expect from this model. So his experiments are really very critically important because we all know, or you should know, that in research, you can only disprove hypotheses. That's what you should be trying to do. You don't try to prove your hypothesis. You try to design experiments to disprove them. And this experiment was designed by Toshio to do just that. And if this number were correct, it would clearly disprove this whole idea because you can't get 60 nanometers of movement from a head that is only 15 nanometers long. Okay? So really important work from the Yanagita lab. But as I'll come to, difficult experiments. And so whether this number is really greater than 10 is the question. So what was absent from the field at this time? Notably absent was the availability of quantitative in vitro assays for the functions of real interest here. Now, I was trained at Stanford in my PhD thesis work by consummate biochemists who taught me that, and all consummate biochemists know this, that you don't even think about trying to understand how a system works if you can't reconstitute that system and the functions of interest with highly purified proteins in the laboratory. And only then can you begin to understand how things are working. And that was really missing from the muscle field. And what are the functions of interest? It's movement and force production. Yes, we had the very important assay of actin-activated ATPase activity, but that's only part of the story. You really want to be able to quantitatively measure movement and force production, which is something I therefore set out to do when I first set up my lab in 1971, and took many years to really establish this, and I'll come to that in a moment. Another notably absent element in this story is we don't know what the high-resolution crystal structure of either actin and S1 are in 1970s, okay? Or even in the 1980s, it turns out, because these turned out to be difficult structures to get. But if you're a structural biologist, you understand that you also are not going to understand how the machine works unless you can, in fact, know what the high-resolution structure of your proteins are. That's absolutely essential to understand how it's going to work. And these were missing in this period, 1969 to early 1980s. And that was a large part of the problem. So after many different kinds of experiments, finally in 1986, a student of mine, an MD PhD student named Steve Krohn, utilized a really important observation, again made by Toshio Yanagita. What Yanagita had shown in 1984 in this paper in Nature, that you could see actin filaments under a good fluorescence microscope if you labeled them with a fluorescent probe attached to a molecule called phalloidin, which binds to the actin and doesn't damage its functional properties in any way. It simply lights up the actin so you can see it. Because without that, the actin filament is only about 7 to 9 nanometers in cross-sectional diameter, and you're never going to see that by regular light microscopy. So that was a really important advance. And what Steve did was use Toshio's advance to put these filaments down onto a surface to which myosin was attached. And in this movie, you can't see the myosin because it's not labeled. But when you put the actin in the absence of ATP, the actin firmly sticks down, as shown here. And then the exciting thing is what happens when one adds ATP to this. And this is almost a real-time movie. And I can tell you when Steve first saw this happening, and I was in my office, he came running in saying, well, I can't tell you publicly what he said. It was a 4-letter word having to do with how excited he was that these things were really moving like this. And of course, he sort of assumed that by the time he got me to the microscope, it would have stopped and I wouldn't believe him, but in fact they were still going and going, and they keep going until finally ATP runs out. So it was really a very important and exciting step, which really was the major part of Steve Krohn's thesis. And having such an assay, as the biochemist knows, now allows you to really explore various things. And so the first thing you want to know is, well, do you need the whole myosin molecule for this to work? And we showed, in fact, you don't need the LMM portion, which has been cut away here, that just the HMM, which has part of the coil tail and its 2 heads, worked perfectly well in this assay. But you can also further subdivide HMM, heavy myromyosin, into a soluble fragment, which is just these single heads that have been freed by another kind of protease treatment. And they also have ATPase activity, whereas this subfragment 2 portion, which is something I'll talk about in my 4th lecture, we'll be highlighting S2, that is soluble but has no ATPase. And in a very important experiment, Yoko Toyoshima showed that all you need for this in vitro motility is this subfragment 1 head called S1. And so the focus became at this point, okay, how does the S1 head work to give you this contractile event? Another really important step, which required, again, no surprise, new technology development, was the need to look at what a single myosin molecule really was doing when it interacted with the actin. Okay? The problem is that all of the experiments that Toshio Yanagita were doing and we were doing and other people were doing to try to understand what the step size really was per ATP required minimally the kind of assay that I showed you that Steve Krohn developed, where you have a lot of myosin molecules on a surface and actin filaments running along the top of them. The problem is, in those experiments, you have to make a guesstimate of how many myosin molecules are really involved in moving that actin filament to then try to interpret what kind of a steps you get per myosin molecule. And that was the difficulty that faced us all in getting the number for what the step size really was. And it became clear, therefore, that someone simply had to look at what a single myosin molecule did when it bound to an actin filament and interacted with it in the presence of ATP. And to do that, we developed this system using biophysics and the physics of laser trapping. And a simple view of the physics of laser trapping is that you can take, shown here by this gray sphere, which, for example, could be a 1 micron diameter polystyrene bead. If you get that bead near a laser beam or the focal point of a laser beam, then because of rules of Newton and Einstein, photons that are being diffracted through this particle and have a momentum upward, in this case, that causes an equal and opposite momentum on the bead, which forces the bead into the laser trap. And the same thing is happening in the reverse direction if the bead is coming from below. So this simple light has momentum and light deflection results in a force is what holds such particles in the middle of a focal point of a laser beam. And then out of a dream, and I'll come back in lecture 4 to my dreams, which my lab somewhat makes fun of, but we'll return to that later because it's another interesting story. The idea was to split a laser trap to bind polystyrene beads, now shown here in purple, that are linked to the ends of an actin filament by ways that we knew how to do for many years. And the idea was, and I call this the simplification of the Steve Krohn motility assay, although you can see from the image below that it's not a simple experiment at all. But it is a simplification in that what we're doing is we're trapping these purple polystyrene spheres, which are attached to a single actin filament, which allows us to maneuver then that actin filament, move it around, and be able to lower it and raise it by focusing and defocusing the laser beams onto other bumps, which tend to be 1.5 micron beads that are glued to the surface of a cover slip. And then the other part of this is that only a very few myosin molecules, very low concentration of myosin, is put into this chamber, so that most of the time there's nothing on the top of the bump. And so what we do is we take this dumbbell of actin and we move it around and we test bumps on the surface, and usually nothing is there, so nothing happens. Finally, you find a bump where statistically there's only one myosin molecule because most bumps don't have anything. And when that actin filament finds that one myosin molecule, you see that in the experiment and you begin recording what happens. And this was done by another MD PhD student, Jeff Feiner, together with Bob Simmons, who was in my lab on sabbatical at the time from England. And what they found was they could measure the displacement of the actin filament by watching the displacement of this bead as it was pulled on by the myosin molecule. The bead is monitored by what's called a quadrant detector, and that's very sensitive to even a nanometer of motion. And so when you're holding the bead with the laser trap with a strength that is well below the strength that the myosin can pull the bead, you get a step size. And that step size turned out to be 10 nanometers. Okay? Furthermore, perhaps I should say that again. 10 nanometers. Not 60, not 100, but 10 nanometers. So while that doesn't prove the swinging Crossbridge hypothesis, it's now consistent with it and doesn't disprove it. But using this same technology developed by Jeff and Bob, you can increase the laser force on these beads, for example on this one, until it just matches the force that the myosin can pull. And when you reach that point, there's no movement that occurs. So it's what muscle physiologists call an isometric tension measurement, but they're used to doing it with a muscle fiber, and here it's being done with a single myosin molecule. And so you, instead of getting a displacement, you get a force readout. And so one can measure the intrinsic force that a myosin can produce, and it's of the order of about 2 piconewtons or so. And that's a number that muscle physiologists have predicted, because you know roughly how many independent force generators you have in your muscle, and if you add all of those 2 piconewton forces up, you get very close to the force that you know the muscle can produce. So this was a very exciting development. Okay, so what about this other really important element that was missing? The crystal structures, the high-resolution crystal structures of the actin and the myosin proteins. Well, this finally came in the 1990s. And first, in 1990, came the crystal structure of actin. Now, actin is termed G-actin or F-actin, depending whether the actin is a monomer. So here's just one actin monomer, sort of spherically shaped here, in the filament. So that monomer is called G-actin for globular actin. And then when it assembles into this two-stranded helical rope, it's called filamentous actin or F-actin. And in really critical papers from the Holmes lab, Ken Holmes in Heidelberg, what they first did was got the first crystal structure ever of the globular G-actin. Okay? And there have been many other crystal structures since then, which are pretty much identical. The other paper shown here below, which also appeared in 1990 in Nature, is an assembly of putting this high-resolution G-actin structure into lower resolution electron micrograph images of the filament itself. And so what you see here in F-actin is a very good model of what F-actin almost certainly looks like. Then in 1993, the crystal structure of the motor domain, subfragment 1, or S1, was obtained. And remember, we already knew from 1987 that this was the motor, but we didn't have the structure. And the structure was really beautiful. It was obtained by Ivan Raymond and his colleagues at University of Wisconsin. And what they found was that in this red domain of the head is the actin binding domain. So think of the actin running up and down vertically here, bound to the head. And about 4 nanometers away from that, about here, is where the ATP binds. And then further down the molecule is a really interesting part of the molecule where the heavy chain extends itself by this blue single alpha helix, and wrapped around that single alpha helix are 2 different gene products called light chains. One's the regulatory light chain, or RLC, and the other is the essential light chain, or ELC. And these stabilize this heavy chain blue alpha helix. And when we and everybody else who looked at this first looked at it, we said, gee, this may be acting like a lever arm to amplify movement that may be a smaller movement happening back here in the head. And so the first experiment that was done to test this was done by Taro Ueda in a paper that was published in 1996. just a few years later. And the experiment was to genetically engineer a myosin molecule with different lengths of this putative lever arm. And what Taro showed is that the velocity in the in vitro motility assay increased linearly as a function of that lever arm length. And presumably because the displacement that you can get from the lever arm goes linearly. Okay. This experiment has been done multiple times, either using velocity as a measure or actually measuring the displacement itself using the laser traps, and you get the same answer. So this kind of experiment was really critical evidence that this neck region, or the light chain binding region, acts as a lever arm to generate movement. Now from 1990s to present, or especially in 1998, there was this critical paper from Dominguez, Friesen, Tribus, and Cohen from Brandeis University showing that they could isolate a different structure of the S1 head that's in its putative pre-stroke state. Okay? And from 1990s to present, there have been a huge number by a large number of investigators looking at a large variety of myosin crystal structures in many different nucleotide binding states, because according to that chemo-mechanical cycle, you should be in a pre-stroke state with certain components in the active site bound, like ADP-PI, and a different state when you have only ADP bound, for example. And so many, many crystal structures were obtained in different nucleotide states, and they were obtained for not only skeletal muscle, but cardiac, smooth, scallop, insect muscle myosin, and even a lot of non-muscle myosins, where a lot of work, especially elegant work out of the laboratory of Anne Hadous at the Curie Institute in Paris, really put the nail in the coffin with regard to this idea of the lever arm swing. So a consensus model for the pre-stroke and post-stroke states is shown in this slide. Notice that the gray head domain with actin binding here at the top has the same configuration in these two pictures. So they're not changing configuration. What's changing configuration is this light chain binding domain going from a pre-stroke state here to a post-stroke state. By the way, around here is where the sulfhydryl group is that the biophysicists were labeling with probes to look for changes in orientation. And of course, it turns out that this part of the molecule doesn't change configuration, and that's why they didn't see any change in configuration. It's only the light chain binding domain that is swinging about some pivot point about here in the molecule. Okay, so here that same swing is shown from a pre-stroke going to a post-stroke state with the head bound to an actin filament, and you'll get about 11 nanometers of movement out of that change, and therefore move the actin filament about 10 or 11 nanometers for one ATP turnover. So the chemo-mechanical ATPase cycle that we now think about is very similar to one I started the lecture with, except the S1 is now depicted having a domain that doesn't change shape, going from the pre-stroke state to the post-stroke state, but only the light chain binding lever arm swings about this pivot point shown by this black dot with the white stars surrounding it. And so we now have a chemo-mechanical cycle where, again, myosin binds ATP, that's hydrolyzed very fast, as Lemon-Taylor showed. And then you're waiting in this state in the relaxed muscle, and when you turn on muscle for contraction, this then can try to bind to actin. That's a slow step. That binding to the actin is the trigger that causes the phosphate release with a concomitant power stroke of about 10 nanometers and leading to ADP release from the active site, so ATP can now bind and release the head. So the summary of this part 2 is, again, you need constant new advances in technology in order to elucidate how muscle works. And in this case, this involved developing quantitative in vitro assays for movement and force production using purified protein systems. The myosin head, or subfragment 1, is clearly the motor domain. The pivotal crystal structures were finally obtained in the 1990s for both pre-stroke and post-stroke states. And so the swinging Crossbridge hypothesis, which is now called the swinging lever arm hypothesis, is widely accepted. So thank you for listening to this lecture, and I hope that you'll stick around for part 3 to come.